Thank you. Thank you very much. It's a pleasure to be here in a like-minded community, so thank you very much for the organizers for inviting me. So I'm going to go super fast. We're interested in genotype-phenotype problem, which means which genes and variants drive which traits and diseases. And we hope to identify them in order to eventually engineer organisms or maybe even cure diseases. Of course, this is extremely hard. Why is that? Well, because DNA encodes function in a very roundabout way through a series of nested network structures, each with cryptic emergent properties. And they are described through mathematical techniques, but they tend to be specialized and niche related, so they don't communicate with each other. So knowledge is split apart as opposed to being lumped together across scales. So what are we to do? We can basically ignore this and carry on. That's fine. We can put all the data in a big machine learning environment and hope for the best. Or I'm going to tell you today about a third path, maybe, which is a theory of fitness. So basically, in this view, you can look at information, at genetic information and life as information diffusing in a fitness landscape. So the problem is to model mutations, the forces they're acting against those mutations and populations. So first of all, we need to establish a vocabulary and some definitions. So what is a mutation? A mutation is something that moves a genome in the fitness landscape. So a mutation is displacement of the genome. What is fitness, however? Well, the ability to reproduce, maybe. More generally, is the ability to carry on the work needed for life, obviously. So high fitness, low fitness. So fitness is literally the potential for work. So fitness is literally potential energy. So in that sense, that's pretty good, because then a fitness landscape can be characterized by contour lines of isofitness, pretty much in the same way that you could have electromagnetic fields with isoelectric lines or gravity fields with isoelevation. And if this is really a potential, fitness potential, then we know that there are forces in the fitness landscape which are defined by the gradient of the potential, and that the work of a move in the fitness landscape, the work of a mutation, would simply be the gradient of the potential times the displacement. So basically, we can even apply higher principles of physics. We can say that in a population, each with multiple moves and a lot of energy from those moves, the energy should be distributed at equilibrium following Boltzmann distributions. So those are predictions that we might be able to verify if and only if we can actually compute a gradient of a fitness landscape and a displacement in space. So how would we compute a displacement of a genome in space? So this dovetails into some of the talks here initially. A displacement is simply a small mutation. And in the case of amino acids, we have a pretty good idea of what the amino acids look like. We know that some transitions are very small and conservative. We know that other transitions are very large and non-conservative. And we can get statistics on that through transition matrices and substitution log odds. So you can easily imagine that you can measure the size of a move from a mutation by looking at the substitution log odds. A move from charged to non-charged would be a big move. A move from alanine to valine would be a small move. So that's, maybe we can do that. Next, we'd like to, however, to get the gradient of the fitness landscape. So in order to measure a gradient of a fitness landscape, you need to develop some sort of metric of fitness. And this is very confusing because I'm fit, you're fit, a fly is fit, bacteria is fit. So how do you measure fitness and differences in fitness as you change genomes? Well, actually, this is well known that in a predator-prey relationship, coevolution exists, so that the sequence landscape of one species is really the fitness landscape of the other species. They are completely mirroring each other. So if you don't know the fitness metric, you can just use the sequence metric on this side, and vice versa. In other words, I'm trying to argue that sequence distances are a really good proxy for distances in the fitness landscape, and therefore you should be able to measure gradients. So let me recap. This is a fitness gradient, which is the perpendicular to the isofitness contours. It's really the ratio of when you have a small move, what is the change in the fitness that you experience. So when you have a small move, a mutation in a protein family, what is the change in the fitness that you see? And as I just argued, that the change in fitness that you see is mirrored by the distance metric in sequence space. So in other words, if a mutation occurs between species that are very closely related, the gradient should be small. If the species are not so closely related, the gradient is higher. If the species are very far apart, the gradient is huge. And you can actually map that on the protein structure. And you can identify areas of very large evolutionary force, high gradient, very low evolutionary force, low gradient. And this actually maps out functional sites very well. There's a huge literature on doing so. So basically, mathematically, I can recap what we're doing. We're doing a mapping from genomes to the fitness landscape. This is the evolutionary function. The evolutionary function hides all of those intermediate emergent properties, which is great. It's unknown, which is not so great. But I just argued that we can actually get the derivative and compute the derivative of the fitness function, which means that if we integrate it back, we should actually get the evolutionary function. We should be able to describe the entire fitness landscape. So basically, I've traded this complexity for three problems. Measuring the gradient of the fitness landscape, I showed you how we can do that. The size of the mutation, I showed you how we can do that. And I haven't yet shown you how we can integrate. So to recap, there's a lot of hypotheses going on into this theory, but if they work out, we should be able to test and use the theory. So again, we measure the gradient of the fitness landscape by looking at which mutations occur between closely related species or very distant species. We measure the displacement of a genome using transition odds. And then we can compute which mutations perform a lot of work and probably are deleterious. And when we do that, we actually consistently perform extremely well against the best algorithms out there to compute the deleterious impact of mutations. This is the KG competitions. It's a blind, objective challenges where nobody knows whether the mutation is deleterious or not. We predict it, and independent judges have experimental results, and we perform systematically, not always the top, but generally quite well against the most sophisticated current state-of-the-art methods to compute deleterious impact. So this is our predictions in specific proteins in a eukaryotic system, in a viral system, in a bacterial system. This is the prediction. This is the experimental impact measured through assays. And this is the prediction. The correlation is not too bad. We can also do that in proteins that are involved in Mendelian diseases. And the prediction then correlates pretty well with the clinical impact. We can even identify thresholds above which p53 mutations are more deleterious to patients and cause higher mortality. And actually, above that thresholds, tumors will tend to be more invasive, have faster growth in vitro, be more metastatic, resistant to cisplatin, but sensitive to a WEE1 inhibitor, and patients who have Leigh Fermanis who are born with p53 mutations. Those which are more deleterious have 20 years less time before they develop their first cancer. So I had mentioned that we can try to see whether the distribution of mutations in populations, ourselves, follows the Boltzmann distribution. So I can take all the mutations, coding mutations you have, score them, and they will follow precisely a Boltzmann distribution. As it's true in macaques, it's true in Drosophila. So basically, in populations at equilibrium, the distribution of the work of our mutations follows the Boltzmann distribution. In mutations that cause disease, however, the distribution is completely non-Boltzmann. And the difference between Boltzmann and non-Boltzmann actually allows us to try to predict which mutations are deleterious in humans. So this leads to two rules that basically in a general population, so this is a multi-scale theory, in a general population, the mutations should be Boltzmann. But in complex diseases or complex traits, the drivers of those traits would be non-Boltzmann. So we can try that in cancer and try to see if we can identify cancer genes. Those are well-known tumor suppressors or oncogenes. You can see that their distribution is really, really, really far from being Boltzmann. So in this way, we can identify or predict genes that may be associated with cancers. And this does better than current techniques to identify genes associated with cancer. We can predict genes that are well known, but also genes that are not so well known. And the genes we predict, which we predict to be oncogenes, actually turn out to be cancer essential genes in tests from the Broad Institute. And for tumor suppressors, we have to do the experiments ourselves. This is with collaborators, the confirmation of a tumor suppressor in head and neck cancer. With other collaborators, the confirmation of tumor suppressor in breast cancer. So we can do this actually even in other complex diseases, not just of somatic tissue, but Alzheimer. And I think, does this work? Yeah, this is the assay we have. So this is a fly assay mimicking a neuropathy due to overproduction of Alzheimer-associated proteins, tau, or A-beta-42. And basically, we can use the techniques I just told you to identify genes that might be associated with Alzheimer. And when we test them in this assay, we get, in this case, half of our genes seem to be validated by this assay. In this other test, 70% of our genes, 68%, seem to be validated. So we've done this not just in cancer or in Alzheimer. We also have data on autism, bacteria, clinical applications for this theory to predict the association between genotype and phenotype. The theory is general, quantitative, computable, multi-scale, complementary to what's known. It allows us to weigh the work of mutations in the fitness landscape and find driver genes. It's based on all those hypotheses coming together. And basically, from a mathematical point of view, what's particularly pleasing to me as a math major is that we can actually do derivatives in the fitness landscape. We can do integration in the fitness landscape. The fundamental theorem of calculus works in the fitness landscape, which you might think is trivial, which I think is kind of not expected necessarily. From a physics point of view, the distribution of our mutations follows the laws of statistical thermodynamics and allows us to predict the Boltzmann distribution of normal population equilibrium, but identify genes that fall outside of this distribution and which drive complex trait to allow us to link genotype to phenotype. Thank you.
So my question is the following. So your main assumption here is that mutations move you around in the fitness landscape. However, that's actually only true for single mutations, right? Because, or if mutations don't interact. Because if mutations interact, you make a move, you change the landscape, right? So is the reason why this approach works so well because you're only looking at single mutations and never look at phenotypes that actually come from pairs of mutations or more?
Right. So certainly, I think that's partially true. The theory is a first-order perturbation theory of the evolutionary fitness function. So we assume we don't look at second-order terms. We don't look at epistasis. So it's just a first-order approximation. We assume independence of the mutations. It would be really nice to have the second-order term, except I don't know their sign, let alone their magnitude. So I think it will take a while before we can introduce second-order term and try to factor those interactions in, which obviously must be very important. It is interesting that even a first-order theory allows us to do some of those computations. And obviously, sometimes we don't predict things perfectly well. And I'm not pretending that this is 100% sensitive and accurate. It's just a first-order approximation that enriches our hypotheses. Yeah.
Hi, thanks. I was just wondering how well the machine learning approach does.
Yeah. So that's really great. So the type of mutations we take into account are coding mutations. We don't take into account non-coding mutations. We do not take into account interactions between mutations. And because I don't know how to develop the theory towards those mutations and those interactions, we can put all this into the context of machine learning. And that's what we're doing right now. Half of the Alzheimer results I presented to you are from machine learning. We're using machine learning on bipolar disease, on myocardial infarction diseases right now. And basically, I think it works very, very nicely. It will allow us to pull into the machine learning context not only the things we know the theory well for, which should be fairly well denoised, but also put everybody else's data, which are pretty noisy, and we hope that the denoised data from evolution can help further denoise the other types of data. So we're very, very much looking forward to having data to show.